So it is recording. So we'll get started here. So thanks everybody for joining. People are going to be joining in as we go, but I'm pretty happy to report that Marie has decided, has allowed us to talk her into giving a talk on BORGs. And I saw the immediate publication on BORGs, and I tried to find the acronym of BORGs, and I couldn't find the acronym, what that meant anywhere in all the articles I read. So, but I'm excited to hear how they play a role with archaea. And Marie, I'll let you introduce yourself and your lab to the group. It's pretty low key, so feel free to ask questions back to us if you'd like to. So it's all yours, Marie.
Okay, awesome, thanks. I'll start sharing my screen and then get started. All right, thank you for the introduction, and thank you for giving me the opportunity to talk about our research on BORGs today. I'm really excited to share this with you, and we'll hopefully have a small discussion in the end to hear your thoughts on things. So the title of my talk is BORGs are giant extrachromosomal elements with the potential to augment methane oxidation. My name is Marie-Charlotte Schoelmerich. I'm a postdoc in the lab of Jill Banfield at the Innovative Genomics Institute of UC Berkeley. And yes, today I'll be presenting to you our preprint that was recently uploaded on bioRxiv, and the manuscript is currently under review at a journal. So I want to tell the story by starting with how it all started. So it all started with the discovery of enigmatic DNA elements in metagenomes. And these enigmatic DNA elements were found across different sites, for example, in vernal pool soil in California, actually in Jill's backyard. That's where we do a lot of sampling. Then at a rifle aquifer in Colorado, also in a Corona mine underground discharge, also here in California. And these enigmatic DNA elements were also found in the East River Shale in Colorado, 50 centimeters below the riverbed. So it was more serendipitous that these DNA elements were found. So we weren't searching for them back then. They just reappeared in our metagenomic data sets. And while these 4 different sites are on the one hand distinct, one aspect that unifies them is that they are all anaerobic ecosystems. So what makes these DNA elements so enigmatic? Well, first of all, they are huge. If you piece them together, which is quite the tedious work that mainly Jill is doing in her free time, we saw that they are extrachromosomal elements that can be up to 1.1 megabase pair in length. And they are linear, terminated by long inverted repeats, and they are always composed of 2 replicas of unequal length. So this is what's depicted on the right-hand side here. So when looking at these pieces of DNA, we started giving them names in terms of colors. So we have a type of DNA that's called black, purple, sky. That color name list is long. We have some very funky color names already. For example, cyan, pink. You'll see a longer list later on. But yeah, what's really striking and characteristic for these pieces is this architecture. So you'll have one large replica. Basically, all the genes are running in one direction. And then you have this other shorter replica, and there all the genes are running in the reverse direction. And one other very striking feature about them is that they are interspersed with numerous perfect tandem direct repeats. So that means that there are pieces of DNA, like words, that follow each other. And these pieces of DNA can follow each other up to 50 times. They can be different in lengths, but while the units remain the same of a region, every region is unique. So you never have the same type of repeat occurring across a genome or even across these DNA elements. So they're always unique, and it seems that they are fast evolving, much like CRISPR-associated repeats, but of course with the distinction that CRISPR arrays have with these spaces in between, and these are perfect tandem repeats. So what makes us think that they are extra chromosomal elements, and why couldn't they be their own, let's say, living entities? Well, first of all, these pieces of DNA always lack ribosomes. They sometimes have one or two ribosomal proteins. So for example, many of them have L11, but not all. For example, that red piece of DNA doesn't have it. And they also lack any known, or mostly don't have any viral or plasmid proteins. There are some exceptions, but they really, when you do protein clustering, you really see they're nothing like anything that we've seen in databases before. But of course, we wanted to figure out what are they. So we looked at the hypothetical proteins that are encoded and found, unfortunately, most of them have unknown domain annotations. So there are a lot of novel proteins encoded. But you can also see that most hits actually match archaeal proteins, and then some match bacterial, and some even match some eukaryotes and viruses. Now, if we look at this even more closely, we see that those archaeal proteins actually almost always match methanoparidins, and more particularly methanoparidins nitroreducens, which are methane oxidizing archaea. So apparently these pieces of DNA are able, well, have DNA that originates from methanoparidins, which is why we believe that they're linked to methanoparidins. Another clue is that in all the sites where we find the BORGs, so the deeper we go in the site, the more DNA elements of this kind we find, and they always co-occur with methanoparidins. So it seems like they have assimilated, so taken up DNA of their putative host, the methanoparidins. And this feature is why we invented the name BORGs for them, because this, if anyone has seen Star Trek, you may know these alien species called BORGs that assimilate things that aren't their own and make it their own to, I think they strive towards perfection. So what does this assimilation look like? If you look on the DNA level, it's quite nice to see it, or it can be seen quite easily if you plot the GC content. So these DNA structures, termed in terms of their color, that we call BORG. So it's a color code and then BORG. So for example, this is lilac BORG. It has a typical GC content of 32%, whereas methanoparidins have a GC content much higher, more than 10% higher, about 45%. So when we looked at the genome structure of these BORGs, we always saw regions that were elevated in GC content. So rather than 32%, it was more close to 40%. And well, not coincidentally, but matching the observation that they assimilate genes from methanoparidins, these gene clusters almost always match the genes that are found in methanoparidins. And this one gene cluster that got us really excited encodes the methyl-CoM reductase, or the MCR, which is the key enzyme responsible for the key metabolism that methanoparidins do, that we'll look at in just one moment. So down here, there's another example of this elevated GC region that really shows that the BORG genome has stretches of genes that originate from methanoparidins, and that's easily seen by this GC content. But we also see sometimes the GC content may be lower, although the gene also has the closest hit to methanoparidins. And we believe that the genome, or that part of the genome, has been ameliorated to match the characteristic 32% GC content of the BORGs. Now, to put all of this into context, why is this important for the methane cycle? Methanoparidins are anaerobic methanotrophic archaea that oxidize methane. So they basically do the reversal of what methanogenic archaea do, which produce methane, and they can oxidize methane back to CO2. Why is this important? Well, methane is a very potent greenhouse gas, about 25 times more potent than CO2, and it's produced by microbial processes, mainly methanogenesis, but also demethylation of methyl phosphonates in the ocean. And as much as 1 gigaton is produced annually by methanogens, predominantly in rice fields, soils, wetlands, and marine sediments. So the efflux of methane into the atmosphere is mitigated by methane-consuming microorganisms, these anaerobic methanotrophic archaea. Then, of course, there's also an aerobic way, but we'll really focus just on these ANME. So methanoparidins are also called ANME2D. They perform anaerobic oxidation of methane, and they couple this to the reduction of extracellular electron acceptors such as nitrate, sulfate, iron, or manganese. And the central metabolism that they use is the reverse Wood-Longdal pathway, which is depicted here on the right-hand side. So you see methane is oxidized to CO2. The electrons travel via multi-heme cytochromes onto extracellular electron acceptors, such as manganese in this case. And there's also some energy conservation going on to make ATP from this process. Now, interestingly, the BORGs have key elements of these methanoparidins that are required to facilitate this type of metabolism. So for example, they have the MCR, we've seen that already, and they also encode a myriad of different multi-heme cytochromes that are required to channel these electrons onto the terminal electron acceptors. In order to really shed light on the metabolic potential of the BORGs, I focused on analyzing the lilac BORG genome really up close. So what you can see here is a cell cartoon of a methanoparidin cell, and everything that's drawn into the methanoparidin cell originates from one BORG, from the lilac BORG. So there's really nothing from methanoparidins drawn in here except for this cell cartoon, well, this cell wall, let's say. So when looking at all the different proteins that are encoded on the lilac BORG, I grouped them into 4 different main groups. So energy metabolism, central carbon metabolism, stress and defense mechanisms, as well as extracellular electron transfer. And I'd like to briefly go over some of the key features of these different categories. So as mentioned, most excitingly maybe was the finding that several BORGs encode the methyl-CoM reductase that oxidizes methane and liberates electrons. But several BORGs also encode a carbon monoxide dehydrogenase that can be used to oxidize CO, and the electrons that are liberated from this reaction can be used for central carbon metabolism. Then there's also an ethyl benzene dehydrogenase-like protein, or a multi-copper oxidase that is most reminiscent to a manganese oxidase. So we speculate it may be involved in manganese reduction. Besides energy metabolism, we also have a central carbon metabolism module that comprises different enzyme sets that are involved in glycan biosynthesis, so cell wall modification, and also polyhydroxybutyrate synthesis, as well as nucleotide metabolism. Then down here in green, we also have different systems tied to stress response and defense. So there are several BORGs contain CRISPR-Cas systems. There's an osmostress response module. So there's a transporter that can facilitate a sold-in strategy. Different compatible solutes can be produced by the proteins that are encoded on this lilac BORG genome. Then there's an oxidative stress response module that can facilitate combat against oxygen, which is of course really important for anaerobic methanotrophs, as they're highly prone to be inhibited or even die when they face too much oxygen in the environment. And last but certainly not least, many BORGs encode multiple multi-heme cytochromes. So lilac BORG, as you can see, has different clusters that I termed, well, 1, 2, 3, 4, 5, and 6. And you can see particularly cluster 3 encodes several proteins with a myriad of different heme binding sites. So we're really envisioning that this can bestow the ability of the methanoparidins that BORG is linked to make use of different redox potentials for routing the electrons that are derived from central metabolism onto extracellular electron acceptors. So as I just told you, BORGs have key enzymes of methanoparidins, and while many BORGs share similar features, they don't all share the same metabolic potential. So this is just a depiction of the metabolic potential of one BORG, but there's, well, way more to be uncovered. So the second set of genes that is, oh, I have not mentioned this yet. So all the proteins that are highlighted in red, we did not find in the methanoparidins that were identified in the same site as the lilac BORG. So that means that either we haven't sequenced deep enough, or we just haven't found the methanoparidins that also contain these genes, or these are actually features that the BORG doesn't share with the methanoparidins. So these are features that are just found on the BORG and not on the methanoparidins itself. So we don't know that for certain yet, but certainly we know something like NRDD should be encoded in methanoparidins if it wanted to, produce nucleotides. So we believe that the genes that are encoded on the BORG genome have the potential to augment metabolism, maybe even substantially, because, well, having isogenes, of course, is a huge benefit to adjust to, for example, fluctuating electron availability or just different environmental conditions, or even like a life cycle. And the BORGs have 1/4 of the methanoparidin's genome size. So that means that they must be tightly regulated, because imagine having, well, this association. So it is archaean with, well, for example, a megaplasmid or a virus or whatever structure the BORG may be. It needs to regulate it. Otherwise, it would just be a waste of energy just transcribing and translating these huge pieces of DNA all the time. Now I'd like to show you a summary table that is a little bit outdated so far. So it shows the different BORGs that we have found at different sites. And our next steps in BORG biology, or one of the main focus now, is for us to find more BORGs, because you know how it is with bioinformatics, the bigger the data sets, the more telling they can become. And so far, we haven't explicitly gone to sites where we anticipate to find them. It was really just coincidental that we found them. And now we're looking, for example, in rice fields. We're going back to Jill's backyard to search more for BORGs and also do some metatranscriptomics, because that would be amazing to get some insights into how BORGs and methanoparidins are linked, to understand their evolutionary history, to uncover the metabolic potential of other BORGs, but also to biochemically analyze interesting BORG enzymes and discover new enzymes, potentially even for genome editing. And with this, I would like to acknowledge the people who did a substantial amount of work on this. So first of all, that's Jill herself, her PhD student, Bassem Al-Shayab, other members of our lab, so Jacob, Louise, Rohan, and Susan, as well as Alex Sisi, who just left our lab to be a postdoc somewhere else. Then our collaborators, Michael Wilkins, Kenneth Williams, Jennifer Doudna, and many others. And I'd like to thank the funding buddies and, of course, you for your attention. And I look forward to, well, hearing your questions.
Thank you, Marie. That was fantastic. I have a couple of questions, and I'm sure there's other people that do too. So, oh yeah, that was really fantastic. It's definitely fun to learn about something new. So, I wanted to ask you a basic question: is how do you isolate these?
Okay, so to get them into the laboratory to actually do wet lab experiments on them?
Yeah, because you got to purify them, right? So when you say they're 1.1 megabases, you have to be very careful in handling and extracting to get them out, right?
Yes. So, but so far we haven't even attempted isolating them. We've only assembled them from metagenomic data. So
Ah,
this data is really coming from short read assemblies where actually next week I'm going sampling to try and recover intact cells. So we can also do some other experiments. But so far we don't have them in the lab, but we have collaborators who have methanoparidins in culture and bioreactors, and we are hoping that somebody will find them in those bioreactors. But so far, it's all from genomes that have been assembled from from reads.
so how are they different from megaplasmids? I'm trying to figure that out here. So there's characteristics of megaplasmids versus your Borgs. How do they differ that really makes them stand off?
Yeah, that's a good question, and I don't have a very good answer for it, other than that they are, well, in terms of their protein inventory, is very different from megaplasmids that have been described so far. We and yeah, also the way that they're replicating, we don't really know yet. We can't really say for certain that they're not megaplasmids or that they're not viruses. It's just that they don't compare to the ones that have been described so
So
far.
you've done alignments against current
Yes.
builds of megaplasmids and whatnot.
Yeah, so a lot actually stems from protein clustering. So we've grouped them to protein families, and they really are distinct from anything that is known.
Interesting. Yeah, I'll be interested to hear when you guys start isolating, long pieces of DNA and running some nanopore
Yes,
sequencing out to see if you can actually find one, like physically find one and isolate it. I'm sure it'll be challenging.
yes, yes. We definitely want to do that, and also to try and link them. I guess there are also other excellent methods using nanopore, as you suggested. So that's what we're looking into.
Questions from the field? Anybody got anything to add or questions for Marie?
Well, I have a question, Scott. So it resembles to some extent the photosynthesis genes found in cyanophages, for example. And if I'm, thinking a bit abstractly in a shorter version, so so do you see a modification in structure, for example, on Borg-hosted MCRs versus native MCRs, something that would give them advantage or higher functionality?
Yeah, that's a very good question that we've thought about a lot already. As you saw from the amino acid identities of the MCR, that they were around 90% identical. It's not something extremely striking, but we do have examples of Borg proteins that are also found in the methanoparidins, but that have been changed on the Borg genomes to that we predict to have a new or additional function. Unfortunately, it's not my topic, so I can't share more on this. So, but I can say that definitely that's something that we're looking into. And we'd also thought about actually producing the MCR enzyme from the Borg and the methanoparidins to get some biochemical data, because prediction is of course nice, but it's even better if you can validate this. And yeah, we're thinking about this, but particularly MCR is so difficult to produce functionally that would require yeah, that will require a lot of work, but that's definitely something we're thinking about. Yeah.
I have a question, or it's kind of a half thought, half question, and it's not exactly what you focused your talk on, but I was thinking about these repeats at the ends, which makes me, of course, think of telomeres. And so I'm just curious, one, have you tried to match them to any sort of telomere sequences, which can be kind of hard because telomere sequences aren't always like a consensus sequence? And then also, do you have any ideas of where these repeats are coming from to be at the ends like that, since I'm assuming they don't have something like telomerase or something to sort of add them. So just curious.
Yeah, we've heard about this a lot. People saying that it's like telomeres. It's also something that jumped in my head immediately when seeing them. So maybe it is a related structure. It certainly has similarity to the telomeres. Absolutely. And we see that every Borg has like its own profile there. So they are not identical, these repeats that are the terminal repeats. We have thought about mechanisms that introduce them, but there are so many unknown proteins, but there are certainly many proteins that have domains that could, in theory, make these telomeres. So we have some ideas which candidates it could be, but we would need to do some biochemistry to validate this. But that's what we're working on with Jennifer Doudna's lab to try and to show this. But I would like to stress that these terminal inverted repeats are very different from these repeats that are found across the genome, and we're thinking that they're not necessarily linked. So these are two repeat types that are distinct from one another, and probably also the way that they are formed are independent of one another. And I'm actually working on elucidating the function and emergence of these repeats that are not part of these telomere-like structures, but found across the genome. So yeah, we're currently doing a lot of work to try and unravel that, and hopefully also unravel these inverted terminal repeats.
Interesting, thank you.
Thanks for the great question.
So I will ask another question. I want to give other people a chance to chime in here. Marie, how, it's risky to do this kind of work with just short read sequences, right? And we avoid it at all costs. So it must make you a bit nervous to work in the area of repeats with short read. And I wonder, is this like paired end data, or is this mate paired end data? Like, how can you be sure? Because like class 3 genomes, you can't do anything with Illumina data. So how nervous do you get, and how confident are you in assembling these repeat structures?
Yeah, that's a great question that we've heard a lot, and it's absolutely true that one has to be extremely careful. And that's also one of the main reasons why we're working hard on getting nanopore long read data. We did paired end reading, and the person who's doing the assemblies is Jill herself. So she manually curates them piece by piece. And given her experience in the field, I feel very confident that the repeats that we see are real. And what we also see in the mapped reads files is actually they're undergoing some evolution. So yes, I'm confident that they're absolutely there. But I do think it's extremely important that whoever does this is very well skilled and has this experience to do these type of analysis. And I agree, it would be amazing to match this with long read data.
Do you think that there's horizontal gene transfer? Like the cyanophages definitely, or a cutting of an element during some kind of reproduction of algae or fungi. But do you think that these are involved in or can be involved in, what do you hypothesize for a horizontal gene transfer of sorts? Is that a path that you guys are looking at eventually, or?
Yes, so we are absolutely interested in this. We definitely feel there's something going on there. So we're thinking that even different BORGs are coexisting within one cell. But I can't really say much more about this at this point, but it's definitely something we're very interested in finding out how they are related to one another and how horizontal gene transfer can be, is involved in their evolution and propagation. And also, like, another thing that I maybe didn't stress too much, but looking at the sheer abundance of the BORGs in these samples, sometimes we see the BORG DNA exceeds the methanoparidans DNA by a factor of 8. And sometimes we see the BORG DNA is only a fraction of the methanoparidans DNA. So that's also something that's really exciting to us to see. Is there maybe a cell that contains multiple of these, or the BORG content of BORG DNA could be much higher than their own genome.
Any questions for Marie from the group?
Yes, about the transfer of the BORGs. Do you have many? Do you have maybe one hypothesis of what would be the mechanism for transfer?
Unfortunately, no, we don't know this yet. I think our first goal is really to show that they exist within the cell. So that's our primary goal at this point, to show this via sequencing. But it's definitely a very interesting question, but there are still so many open questions tied to them that it's very hard to make the call. What are the ones that we're targeting right now? But it's definitely on our list.
Thank you.
Thank you.
So what's your lead time for doing some nanopore stuff on this?
You mean when we plan to do this?
Yeah, start searching for them physically.
Yeah, well, we had planned to do it last week, but because of COVID, unfortunately, we had to change our plans and delay our sampling trip to next week. And as soon as we have those new soil samples, we'll start extracting and work on that. So it's coming up shortly.
And where have you submitted your bioarchives paper?
At Nature.
Okay, good luck.
Thank you. Yeah, I think Jill has been invited by several journals. We were really surprised and amazed by all the attention that this preprint got, even before publication. So yeah, it was really exciting that a lot of people are excited about this. And thank you for inviting me to present our work.
Yeah, I was one of the ones that saw the preprint right away and wrote to Jill right away. So thanks for agreeing to give a talk. Is there any more questions for Marie? Anything that you guys want to comment on, or?
If there's still time, I'm curious about the bioinformatic analysis. So, I mean, do these BORGs form an independent bins in the binning process, or how do you isolate them from their hosts or from the methanoparidans?
Yeah, that's a great question. Yes, they do form independent bins. So a nice aspect is that they have a very characteristic GC content, which is about 32%, whereas the methanoparidans have a 45% GC content. So that's one way of binning them. And the other way is actually by the repeats, because they're so characteristic and they're found in so many sites and these repeats are not found on any other context, actually. So that's a pretty nice way of binning them.
You don't have any idea on potential hosts or where This could be right. Or
Yes,
did I
they
miss
are.
it?
We believe that their hosts are the methanoparidins because the majority of genes that have resemblance to anything that's out there are methanoparidins, and also the methanoparidins are in the same site. So whenever we find BORGs, we also find methanoparidins. So it's co-abundance patterns and the fact that they've assimilated these genes that tell us that they must be linked.
Can it not be mobile elements that have some methanoparidins genes that are hosted by other organisms, similar to cyanophages, for example, if we go back to the same example? I mean, if I go back to the graph that you showed about the classification, in fact, the majority of the proteins were unknown and unidentifiable, and whereas the largest part of those that you could identify were archaeal or methanoparidins. So this is not an absolute link, right?
Yes, it's absolutely true. It's not an absolute link, and that's why we're doing some sequencing technologies to try and link them to unequivocally show this. I think that's going to be important. So far we don't have that yet. It's just these, and it is possible that they're mobile genetic elements. We can't rule that out either. So that's why it's also really important for us to find them, find more sites where they are found to get better statistics on this too.
Thanks.
Any more questions? Cool. Thank you, Marie. That was fantastic. I'm
Thank
sure
you so much.
we'll be hearing more about this, no doubt about it.
Yeah.
So I'm going to move on to a little bit of a spotlight on some technology here. Mark Brown is here from Claremont BioSolutions. I'm not sure if people are aware of them. We got introduced to Claremont Bio from some of the stuff that's happening on the International Space Station. And I will let Mark take the screen and give you a 10 or 15 minute rundown of what Claremont's up to and some of their cool portable technology that you guys might find useful for field work. So it is up to you, Mark, and see you already got the screen.
Thank you very much, Scott, and thanks for the invitation. So at Claremont Bio, we're a sample prep company. We specialize in rapid lysis and nucleic acid extraction from even hard to lyse samples. And so we really got our start after 9/11 and strange envelopes with white powder being shipped around when Department of Homeland Security started the BioWatch program. And they wanted to be able to have an air monitoring system to look for biothreats that was live. So after BioWatch 2, after BioWatch 1, they started BioWatch 2, and we were part of a work project for that. And we were supposed to develop a method that would allow continuous mechanical lysis of hard to lyse organisms. Really, it was to be able to pop anthracis spores if they were collected and moved right downstream into monitoring. And so this is an example of Bacillus subtilis spores that have been unprocessed. And after passing through our rapid lyser, we were able to lyse most of the spores. And there seemed to be some that were still intact, but when we looked at these closer, what we found is that they'd actually been popped and they were leaking the contents, as you see this very stringy material coming out. In comparison to other mechanical lysis methods, in this chart here, we have rapid lyser in blue compared to the BioSpec bead beater, which was the most used and leading competitor in the field at that time in 2009. We were very fast, almost 6 times faster than that, and it could be used in line and continuously. Well, the BioWatch program didn't survive Congress's budget cuts, but we realized at that point that we had a very effective method for lysing difficult to lyse samples. And so we wanted to make something that people could take into the lab, into a biological safety hood, or into the field. And so we developed something called OmniLyse. And in green here on this chart, it's showing the use of OmniLyse to lyse Bacillus subtilis spores. And this was our prototype. And so this is our OmniLyse device. It uses a micromotor, a specialized impeller and beads, and it allows the sample to flow back and forth through it. It goes about 40,000 RPMs, which is a lot faster than you would typically get from a bead beating machine. And because of that, it generates tremendous shear forces. And as the beads pass by each other, these shear forces rip open the cell, releasing DNA, RNA, protein. On the right is just an EGL analysis of a lysate, just showing some of the genomic DNA and the ribosomal RNA that you could get from a sample. Now, not all samples are single cell samples, easy to work with samples. So we've taken this technology and we've modified it so it can be used with plant or tissue samples, or samples that can't necessarily flow through, such as stool or some other sample. And all of these devices can be used with the battery pack, making them very easy to take with you wherever you need to work. So since that time, we've been working on optimizing this technology to effectively lyse the complete sample. And so for most bacterial cells and spores, we typically tell people to run the sample for 3 to 5 minutes. For larger, hard to lyse microorganisms like yeast and fungal cells or parasite cysts, we say 5 to 10 minutes, and then you'll have a very good lysis of your sample. And this is just EGL analysis of different types of samples that are hard to lyse. This is spores, giardia that are concentrated from a water sample, yeast cells, mycobacterium, tuberculosis in a sputum sample, and bacteria from a stool sample. And in each case, we were trying to highlight that we could isolate high molecular weight DNA from these samples. What's nice about our technology, because it's flow through, is it doesn't suffer from overheating, which you can get with bead beating, because we're not introducing new air into it to generate heat. OmniLyse can be used with a number of different extraction kits to improve nucleic acid. However, we don't suggest anyone use them with trizol or phenol chloroform, since those organics can actually etch the plastic that they're in. So how effective is OmniLyse? And so I'm going to show you some of the work that was completed by outside research groups. This is the USDA, and they took 12 extraction methods, looked at cultured foodborne bacteria, and compared the simplest from boiling water up to an automated system on how effectively it could lyse and extract DNA for 16S rRNA copy number evaluation. What they did is they compared all 12 methods and then took the top 3, and we were one of them, and then compared 15 additional foodborne bacteria isolates using these 3 methods. There's a lot of information in here, but this is Claremont BioSolutions results. And in every area where it's green, it's showing where we were in first place in the amount of DNA that was extracted. And then second in pinkish orange, and third in yellow. But in each case, where we weren't first, we were very close typically to what you could get in terms of the amount of DNA that was extracted. But the take home from this was to essentially say that you could provide near quantitative extraction of genomic DNA from a sample using this technology. Additionally, by using OmniLyse, you can substitute other forms of mechanical lysis to improve extraction of DNA. And this is from the FDA and CDC. They were investigating cyclospora infections in pools, and they were using the ZR fecal DNA mini-prep kit. And what they found is they were not able to extract enough DNA to go downstream to do NGS sequencing using the FastPrep 24, but when they used OmniLyse, they were able to increase the amount of DNA they were able to get, and then use that for next-gen sequencing. Additionally, this is one of the posters that Oxford Nanopore put out a few years ago, and they wanted, with their new rapid library prep kit, they wanted a fast method to be able to identify different types of microbes. And so they used OmniLyse during the cell lysis step that went downstream into a purification step, library prep, and then fast identification using WIMP. And what they said is they found that we were very effective at rapid lysis and could maintain large fragment size, and that it worked with a large range of microorganisms. However, because this was lysis, you needed a purification step because to clean the sample up enough for nanopore sequencing. Now, in addition to lysis, we've been working on nucleic acid extraction. And so our first attempt at this was our PureLyse kit. We use a set of proprietary buffers with our OmniLyse. It allows you to do lysis and extraction in a single step, wash away the cell lysate and most of the protein, and then collect your DNA in a third step. And all of this can take place in less than 6 minutes. Now, back in 2010 to about 2015, most of the groups we were working with were looking for rapid identifications of pathogens, either by PCR, isothermal amplification, or lateral flow. And so this was really a good method for bacterial pathogens, being able to quickly get a sample and get DNA out that would be compatible with these different things. And so this is just showing PCR with Mycobacterium tuberculosis that was isolated from a sputum sample compared to the Halsey method, which was the standard method. And we could do this in about 6 minutes. This is a slightly modified pure lyse protocol since it worked with Trichomonas vaginalis, which is a parasitic protist, but you could detect down to 10 copies of T-vag in a sample. And then another nice thing about our technology is because it is so small, you can integrate it into a cartridge in this example here. And this was a cartridge that Angelica Niemes from KGI was developing to be able to detect tuberculosis in the field, and it included sample prep, isothermal amplification, and lateral flow detection. And it was after this that we got our first introduction to extreme microbiomes, and that was through an interaction with the Search for Extraterrestrial Genomes, and that was working with Chris Carr out of MIT, who was working in Maria Zuber and Gary Rufkin's group. And they wanted to be able to develop a cartridge that could go on the Mars rover and be able to test for life on Mars. And so they realized that they could integrate our OmniLyse technology very easily and do an extraction of DNA. And this paper, which is by Angel Maharo, who is pictured at an extinct volcano doing an extraction, was looking at Mars analogs to see if they could extract Bacillus subtilis spores DNA and then go downstream to sequence it on Oxford nanopore minion. And so this was presented at the 2016 NCM meeting by Julie Hatchie, and they were able to extract DNA from a variety of different samples and sequence with as little, I believe, as 10 picogram. Now, the difference that they had to, one thing that they had to do is our method wasn't developed for very acidic or very salty samples. So they had to use a desalting step and a hexamer step to allow recovery of the DNA from the sample types, but they were able to recover enough samples for sequencing. Our second introduction into extreme sequencing or extreme sample collection was working with NASA Ames. They had a project to test gene expression in space on the International Space Station. And they needed a extraction method that didn't use toxic chemicals and could be integrated into a closed system that would allow them to extract up to 20 micrograms of DNA from a variety of samples, and the RNA, and the RNA was high quality. And so we developed a fast flow, low pressure column called RNA Express here that was attached to the OmniLyse device. So you would lyse your sample, bind your RNA, and then collect your RNA. And this was the sample Wet Lab 2 sample prep module that the engineers developed and optimized with our technology. And using that, they did the first gene expression testing in space. And this is just the Wet Lab 2 sample prep module and all the components that they needed to do before going on to the Smart Cycler. And this is Jeff Williams over here on the bottom right, one of the astronauts who was prepping the samples in a glove box on the International Space Station. This was in 2016. And so this paper is in PLOS One and Macarena Pira. So we used the RNA Express protocol that we developed, and we've since gone on to test it with a variety of different samples. We can use it to isolate high quality bacteria from culture in under 20 minutes with RQI or RIN numbers that are greater than 9. We've used it for live dead testing of M. tuberculosis in cerebrospinal fluid samples as part of a collaboration with Jerry Cangelosi from the University of Washington. And we've done some applications with COVID-19. We've also been able to use this technology to improve extraction of degraded RNA from formalin-fixed paraffin-embedded samples. And that's just down here on the bottom, where in comparison to QIAGEN, we were able to get anywhere from 2 to 4 times more RNA extracted from a small FFP blood than using the QIAGEN kit. And what was interesting was we were able to also get much longer RNA. And so we worked with collaborators at CSUN, Jonathan Kelber's lab, and they were published a paper in OncoTarget using some of our methods to be able to improve RNA extraction from tissue samples. So in addition, we're very interested not only in RNA, but isolating single cells or single cell sequencing, but also high molecular weight DNA. And so some of our technology
Hi.
we've used.
Hi. I have to return him.
Okay.
No, his behavior is a safety concern.
And so you can take a small piece of live tissue and put it into one of our devices, and we're able to generate highly intact single cell suspensions. And we've used this to go downstream to grow the cells in culture to isolate specific types of cells. And in collaboration with a company called IncellDX, they've been using it for non-small cell lung cancer diagnostics to identify tumor samples. Now, our hope is to use this technology, since we can get very intact single cells, to do single cell assays. And so we haven't been able to do that yet, but we're hoping to do that in the future. But once we were able to get intact single cells or nuclei from frozen samples, we realized we may be able to get high molecular weight DNA. And so down below here is our DNA Express protocol, where you can take a frozen or a live sample, you can very quickly disaggregate it in our OmniLyse X, and then digest it to get very large DNA. We can put that over a fast flow column, and then we're able to isolate high quality, high quantity, high molecular weight DNA. And we've been able to improve the technology so that we can improve the size of the DNA to get very long megabase sized DNA using this technique. And this is a pulse field gel electrophoresis showing the spread of the DNA using different techniques. We've also looked at the DNA using a technique called molecular combing, which allows you to stretch out the DNA on a silanized cover slip, kind of like optical mapping, and then label it with yo-yo 1. This white bar at the bottom of this image is about 50 kb in size. And what you'll see is we have very long DNA stretched across the cover slip. And it can be used for fiberfish analysis. Then lastly, we've been able to use these techniques and move them to more difficult to work with samples, such as gram-positive bacteria. This is B. subtilis. And we were able to get megabyte-sized DNA. And by using a combination of enzymes with our lysis, we were able to take the Zymo community standard, which is a mixture of 8 bacteria and 2 yeast, and get a decent size sample between about 30 to about 350 base pairs in size using this pulse field and gel analysis. Now, it's one thing to get the DNA. We also have to make sure that it's compatible with long-read sequencing. And so as part of an NIH-GRI phase 1 grant, we were able to isolate DNA from blood and liver samples. That's down on the table below in 4 through 7. And this was done in 2019. We were able to get, using LSK 109, reasonably sized N50s and decent sized output, comparable to what was currently being done in the field. Using John Tyson's method, the Rocky Mountain DNA extraction method, we applied that with colorectal tumor samples, and we were able to generate high yield data sets. Although our N50s weren't very large, we were able to get reads over 100 kb and long reads. So we're pretty excited about that. So in addition to being able to work with blood and liver, blood and tissue samples, we also wanted to get back to what we really have liked to study, and that's the identification of pathogens. And so one of the things we want to do is, can we use our technology to really quickly extract samples from vectors and be able to identify pathogens? And one of the samples that we worked on were the deer tick. And so this example of the Pacific deer ticks, we took 4 of them, we put them into our microhomogenizer with beads, lysed them, collected the lysate, purified over DNA Express. This took about 30 minutes, and then we were able to move downstream through Rad4 sequencing. And very quickly, one of the first sequences that popped in WIMP analysis was Rickettsia. And so it was a very quick method to be able to identify a vector-borne pathogen So we were pretty excited about using it for that application. And we tried it with 2 different flow cells. One was a used flow cell in ion that had previously been used for mouse liver, but we were able to get about 767 hits. And then using a fungal, we were able to get about 1,400. So it was very, very good at being able to identify them, and we could start to piece together the rickettsia genome.
Looks like Mark disappeared. That's what you call clicking the wrong button, right? So until he logs back on, I will just let people know that. So how these work, just so you know, and I didn't want this to turn into commercial, it's just a tube or a syringe, and you put your sample in it, and there's this little wire that comes off, and you plug it into a power pack and turn the on switch on. It's no bigger than like a pack of cigarettes, and you turn it on, and it just buzzes at extremely high speed and just rips things apart. So you can bring them out in the field, and they got these little battery packs. So you can just bring a whole bag of these tubes and a battery pack, and you extract your sample in the field. So the next time you go to Antarctica, JJ, you could grab one of those, or Nicole, when you're down there. We have the variety pack, and it seems like it has some applications. So it looks like Mark might have just hopped back on, and if
Yeah,
we've got some time for some questions, we're going to wrap it up here.
okay.
If you have some time for questions, we're at 4:29. You mind taking a few questions, Mark?
No, I don't mind. I'll just skip to the end real quick, one slide. We're automating these processes to do different things. And our hope is to do total nucleic acid extraction, high molecular weight DNA extraction from a variety of samples. And this is just an example of our automated cartridge, which will go onto our simple prep instruments. We have some for in the lab and one that will go in the field. And this is just to show the high molecular weight DNA jelly that we're able to get and then move downstream to extraction. So I'll take any questions.
Mark, do you have a video real quick that shows this in action?
I have, go back here. I can show it processing. So this is a stool sample that's been put into the processing chamber. You have a processing chamber, the reagents for the extraction, and a column inside the cartridge, which binds and then gets washed and dried. And so this is it, processing the sample with beads. We don't have a full video for this. It takes about 20 minutes to 30 minutes, depending on what type of sample you're working with. So, but that's something that we could post if people are interested.
Yeah, so what I would say, because this is an Extreme Microbiome group, we're talking about going in the field, right?
Mm-hmm.
And, you know, like some of us, you go to bring one of these with you in the field, you plug in, there's a power pack and turn it on and extract it. And then you have to do a purification step, I assume. And do you have a field purification step that you guys are linking to this? Have you gotten to the point of the purification step yet?
Yes. And so this is what we're hoping to take out in the field. This is our FX1 device or system. It allows you to do one at a time. And it has an 8 to 12 hour battery pack, so you can run multiple samples. And it's in a weatherproof case and totally rechargeable. And our hope, one of our first samples to do is some marine filters or samples that are on a filter. And this is an example.
Filter, have you guys done a filter with the OmniLice? I don't know if it's the OmniLice or what you call the one where you have a 2 mil tube with the vortexer on the bottom. Have you done filters with those? Do you have to add something that shreds them up?
So we have done that with our OmniLice X. That's the 2 mil tube. This is an example of nanopore downstream of extraction from a 25 millimeter PVDF filter that we got from Nathan Trulove from Ambari. And this is a gel extraction from Simple Prep, see genomic DNA and what we believe is RNA from the sample. And so we were able to do that. You put the filter in, you add beads, and then you run it for 2 to 3 minutes. You then pull that off, and then you can go downstream into the extraction method you want. Cool. And so we've been putting it into our DNA Express, and we were able to isolate about 2 micrograms of DNA from that single filter, and then go downstream into RAD 004 rapid library prep, and then put that onto a fungal. And then this is some of the results from that test. And we're pretty excited. We were able to get DNA viruses, archaea, a variety of different ocean bacteria, and we were able to get algae, marine mammal DNA hits, so a variety of different samples.
So does anybody have any questions or comments for Mark? I know we're kind of getting toward the end here, but.
How much work have you done with like different soil types?
No, that's a great question. So we've been working on soil for a few months now. We're able to extract high molecular weight DNA, but we're not happy with, we're getting some contaminant carryover that we have to resolve first before it's something we're willing to release. But that's something we're definitely interested in, similar to what we're doing with the stool samples.
Okay, thank you. And if you ever need more ticks, we have lots of ticks that we can send you. So
yes, please. That'd be fantastic. So yes, I'd be very happy with that.
All right.
Yeah, and also, if there's anyone who has a sample that they have an issue with, if they're interested in us trying it, there may be a possibility we could try prepping a sample for you. It gives an idea, since we haven't had much experience working with extreme microbiome samples, the opportunity to try something.
Yeah, one of the things, Mark, is I would say probably most everybody on here would agree that the amount of material that we get from a lot of these samples is very tiny amounts of DNA or RNA. And, you know, RNA is definitely something we all work on. But, you know, it's not uncommon for us to get 3 nanograms or 8 nanograms or something like that. So when you show this long DNA, for me, I don't know when I've ever had a sample that had more than, you know, 30 nanograms. So
yeah, no, you mentioned that before, and we've worked with samples where we're getting about 10 nanograms back, but we've always been too chicken to try and put that on a nanopore.
You can. I mean, what you put on the nanopore for RAD 004 is exactly what you expect. So if you put on, as you know, 6 nanograms, you're going to get that number of reads back. If you put on 50 nanograms, you'll get that number of reads back. So we've done titrations down to 500 picograms with RAD 04, but you're just going to get that number of molecules back because it's just, you put on, you know, it's basically a little molecular pump. So
yeah.
Any more questions or comments by the group before we wrap this up? And yeah, I will send Mark, your email should be on the last XMP email that I sent out. So yes. Krista just said she wanted, and you can kind of share with people that contact you what kind of you know, how people buy them. We got the variety pack, which was really a good idea, and the stuff's really cheap, so it's not going to make you bankrupt by any means. So having said that, I just want to mention next month, I'm making arrangements with John Priscu up at Montana to give a talk on salsa, and some of you may or may not know John. He's pretty well known in Antarctica for doing the deep drillings into subglacial lakes that are quite deep. similar to Vostok. So he's going to give a talk. He's agreed next month on the salsa project, and I'm pretty excited about it because the stuff that he's been doing in Antarctica and up in Greenland with these deep drillings is really super fascinating. I wish I ended up working for him, and it would be fun to freeze to death drilling deep into subglacial lakes. So he's going to be here, and we're going to probably have one more or two more speakers, a technology spotlight. If there's certain things you guys want to hear about as technologies, I'm happy to recruit them to give a brief talk. But other than that, anything else that you guys want to go over? Well, we got the group on the line, or you want to end this now and talk next month? Open format. I'll take that as we're wrapping it up for the day, and I'll say thanks to Marie and thanks to Mark and thanks everybody for attending, and we'll see you in a month.
Thank you very much.
Thank you.
Thank you.